Hello everyone and a huge welcome back to MooTechie. We're so glad you're here. Today we're diving beneath the waves to explore a topic that is both fascinating and crucial for food safety, the microbiology of fish and other seafood. This is a world of delicate flesh, rapid spoilage, and constant battle against unseen microbes. So let's cast our net and see what we catch. Now, let's begin our presentation. Microbiology of fish and other seafoods. Fish is a wonderfully nutritious but highly perishable high-protein food. It typically contains a high level of free amino acids which are like a ready-made meal for microorganisms. The lipid content can be quite high, up to 25%. A key difference from meat is that fish has very little connective tissue, only about 3% of its total weight, compared to around 15% in meat. Generally, fish flesh contains 15 to 20% protein and less than 1% carbohydrate. We can split fish into two categories. Non-fatty fish like cod, haddock, and whiting have fat levels of only about 0.5%. In contrast, fatty fish like mackerel and herring can have fat levels varying dramatically between 3 and 25%. Let's look at the general composition of a fish. It's made up of 65 to 80% water, 1 to 20% fat, and 14 to 20% protein. Now, let's examine a detailed table of the chemical composition for various fish and shellfish. Starting with bony fish. Bluefish is 74.6% water, 0% carbohydrates, 20.5% protein, 4% fat, and 1.2% ash. Cod is 82.6% water, 0 carbohydrates, 16.5% protein, 0.4% fat, and 1.2% ash. Haddock is 80.7% water, 0 carbohydrates, 18.2% protein, 0.1% fat, and 1.4% ash. Halibut is 75.4% water, 0 carbohydrates, 18.6% protein, 5.2% fat, and 1.0% ash. Herring is 67.2% water, 0 carbohydrates, 18.3% protein, 12.5% fat, and 2.7% ash. Mackerel is 68.1% water, 0 carbohydrates, 18.7% protein, 12% fat, and 1.2% ash. Salmon is 63.4% water, 0 carbohydrates, 17.4% protein, 16.5% fat, and 1.0% ash. Swordfish is 75.8% water, 0 carbohydrates, 19.2% protein, 4% fat, and 1.3% ash. Now, for crustaceans, crab is 80% water, 0.6% carbohydrates, 16.1% protein, 1.6% fat, and 1.7% ash. Lobster is 79.2% water, 0.5% carbohydrates, 16.2% protein, 1.9% fat, and 2.2% ash. And finally, mollusks. Clams are 80.3% water, 3.4% carbohydrates, 12.8% protein, 1.4% fat, and 2.1% ash. Oysters are 80.5% water, 5.6% carbohydrates, 9.8% protein, 2.1% fat, and 2.0% ash. Where does contamination come from? The overall sanitary quality of the water where these animals live is absolutely key to the microbial quality of the final product. But it doesn't stop there. Microbes are also picked up during various processing steps like peeling, shucking, evisceration, and breading. Now let's talk about the inevitable process of spoilage. Fish can be spoiled by autolysis, which is self-digestion by its own enzymes, by oxidation of its fats, or by bacterial activity. Often it's a combination of all three. Fish flesh is actually more perishable than meat. This is because of rapid autolysis by powerful fish enzymes, and because its flesh is less acidic, which favors microbial growth. Furthermore, the unsaturated oils in fish are more susceptible to going rancid through oxidation than animal fats. Rigor mortis, the stiffening after death, is hastened by the fish struggling, a lack of oxygen, or warm temperatures. It is delayed by a low pH and adequate cooling. Spoilage really begins after rigor mortis passes, when juices are released from the flesh. All foods have their own distinctive microbiology. For seafood, this complexity comes from specific contamination from the environment and during processing, and from growth conditions created by factors like temperature, pH, and microbial interactions. Initial contamination is due to the wide range of environmental habitats, from freshwater to saltwater, and from tropical to Arctic waters. The variety of processing practices, from simple iced fish to sterile canned products, also plays a role. The part of the microflora that will ultimately grow is determined by the fish's own properties and its storage environment. Several intrinsic factors in fish greatly influence its microbiology. These include its poikilotherm or cold-blooded nature, a high post-mortem pH, usually above 6.0, the presence of large amounts of non-protein nitrogen, and the presence of a compound called trimethylamine oxide. Bacteria establish themselves on the outer and inner surfaces of the live fish, like the gills, skin, and intestinal tract. The fish's cold-blooded nature allows bacteria with a broad temperature range to grow. The microflora of temperate waterfish is dominated by psychrotrophic gram-negative rod-shaped bacteria. Key genera are Pseudomonas, Moraxella, Acinetobacter, Shewanella, Flavobacterium, Vibrionaceae, and Aeromonadaceae. Gram-positive organisms like Bacillus, Micrococcus, Clostridium, Lactobacillus, and Corynebacterium can also be found. The flora on tropical fish often has a slightly higher load of gram-positive and enteric bacteria, but is otherwise quite similar. An absolutely critical intrinsic factor is the very high post-mortem pH. Most fish contain very little carbohydrate in their muscle, so only small amounts of lactic acid are produced after death. This high pH allows pH-sensitive spoilage bacteria like Shewanella putrefaciens to thrive. The non-protein nitrogen fraction of fish is a readily available bacterial growth substrate. It consists of compounds like free amino acids. The decomposition of sulfur-containing amino acids like cysteine and methionine is particularly important, as it causes terrible off-odors and off-flavors due to the formation of hydrogen sulfide and methylmercaptan. Now, what factors influence the kind and rate of spoilage? First, the kind of fish matters. Flat fish spoil more readily than round fish because they pass through rigor mortis more rapidly. However, a flat fish like halibut can keep longer because of the low pH 5.5 of its flesh. Fatty fish can deteriorate rapidly due to oxidation of their unsaturated oils. And fish high in trimethylamine oxide soon yield appreciable amounts of trimethylamine, which gives that classic stale fishy smell. The condition of the fish when caught is also vital. Fish that are exhausted from struggling spoil more rapidly, probably because they have less glycogen, leading to a smaller drop in pH. Feedy fish, those with full intestinal tracts, are also more perishable. The kind and extent of bacterial contamination is another major factor. Contamination occurs from mud, water, handlers, the fish's own exterior slime, and its intestinal content. This can happen in the net, on the fishing boat, or on the docks. And finally, temperature is arguably the most important factor. Chilling the fish is the most common method for delaying spoilage. Cooling should be as rapid as possible, down to 0 to -1 degree Celsius, and this low temperature must be maintained. The warmer the temperature, the shorter the storage life. Now, how do we fight back? How do we preserve fish? Of all flesh foods, fish is the most susceptible to spoilage. Evisceration should be done promptly to stop active digestive enzymes. Rigor mortis itself is a form of preservation, as it retards autolysis and bacterial decomposition. Lengthening rigor mortis lengthens keeping time. Aseptic methods are difficult to apply, but gross contamination can be reduced by cleaning and sanitizing boats, decks, and equipment. One powerful tool is the use of heat. Live crabs are cooked in retorts at temperatures up to 121 degrees Celsius to facilitate meat removal. Canned crab meat is typically pasteurized at 85 to 87 degrees Celsius and then preserved by refrigeration. Most other canned seafoods are heat processed to be commercially sterile. However, seafoods are low-acid foods with a slow rate of heat penetration, making them difficult to process safely. Another cornerstone is the use of low temperature. Chilling is a temporary but essential method. Fish autolyze and fats oxidize rapidly at warm temperatures. Chilling is required from the catching area all the way to the processing plant. Small fish and processed fish are often more perishable. Fish are typically stored in crushed ice, sometimes with added preservatives. Freezing is a step further. In the past, ice with added salt was used. Now, mechanical refrigeration and sharp freezing are standard. Fish are often glazed with a layer of ice for protection. Quick freezing is better for preserving the quality of fillets or steaks. However, freezing is not a sterilizing process. It kills some microorganisms, but many survive, ready to grow again upon thawing. Fish carry a flora of psychrotrophic bacteria like Pseudomonas and Acinetobacter that survive freezing. Worryingly, spores of type E Clostridium botulinum can survive and produce toxin at temperatures as low as 3.3 degrees Celsius. Other preservation methods include irradiation, though it's not widely practiced for seafood. Preservation by drying is a traditional method. Dry salting or immersion in brine works by removing or tying up moisture. However, oxidation of fish oils is not retarded and can still cause deterioration. Salt cod is prepared by a combination of salting and air drying. Smoking also results in drying. The use of preservatives is common. Salting is effective both for drying and for the antimicrobial effect of sodium chloride. Fish may be dry salted to contain 4 to 5% salt. This environment selects for halophiles which can discolor the fish, for example, causing a red color from Serratia salinaria. Other chemicals like benzoic acid, sorbic acid, and nitrites have been used with varying success and legal permission. Antibiotics like chlortetracycline have been used as a dip or in ice. Storing fish in an atmosphere with increased carbon dioxide can also lengthen its keeping time. Pickling involves salting or acidification with vinegar, wine, or sour cream. Herring, for example, is often salted, spiced, and acidified. Smoking was formerly used primarily for preservation, but now with canning, chilling, and freezing available, much smoking is done primarily for its delicious flavor. Fish to be smoked are usually eviscerated and may be salted first, which also improves keeping quality by reducing moisture. Smoking can be done at low temperatures or high temperatures that partially cook the fish. How can you tell if fish has spoiled? The change is gradual, making it difficult to pinpoint the exact start of spoilage. Practical tests like the trimethylamine test for saltwater fish or estimates of volatile acids or pH are used. Bacteriological tests are accurate, but often too slow to be useful. There is a succession of external changes in a spoiling fish. The bright colors fade to dirty yellow or brown. The slime on the skin increases. The eyes sink and become cloudy. The gills turn from pink to a grayish yellow. The flesh softens and exudes juice. A sequence of odors evolves. It starts with the fresh, seaweedy odor, then becomes sickly sweet, then stale and fishy from trimethylamine, then ammoniacal, and finally putrid from compounds like hydrogen sulfide and indole. Cooking will unfortunately bring out these odors even more strongly. Finally, let's look at the spoilage of specific types. Salt fish are spoiled by salt-tolerant bacteria like Serratia and Micrococcus, which can cause discolorations. Molds are the chief spoilage organisms on smoked fish. In chilled shrimps, Acinetobacter, Moraxella, and Vibrio are responsible. Crab meat is deteriorated by Pseudomonas and Proteus. Oysters remain good as long as they are alive in the shell but decompose rapidly when dead, as in shucked oysters. At chilling temperatures, spoilage is termed souring and is caused by Pseudomonas and related genera. At higher temperatures, souring can be from fermentation by coliforms or lactic acid bacteria. An uncommon spoilage by yeast can cause pink oysters. The bacteria causing spoilage are the natural flora of the fish. At chilling temperatures, Pseudomonas species are most likely to predominate. These bacteria break down the fish's compounds, producing trimethylamine, ammonia, foul-smelling amines, and eventually hydrogen sulfide and indole, the hallmarks of putrefaction. A musty or muddy odor can come from Streptomyces species in the water. Discolorations can be caused by various bacteria, a red or pink color from Sarcina or Micrococcus, and a chocolate brown color from yeast. And that brings us to the end of our deep dive. Thank you. We've journeyed from the living fish in its aquatic environment to the complex microbial battle that determines its shelf life on our plates. We've seen how factors like temperature, pH, and contamination converge, and explored the many ways we try to outsmart spoilage, from freezing and canning to salting and smoking. Understanding this delicate relationship is essential for delivering safe, high-quality seafood from the ocean to the consumer. Thank you for your attention, and for joining us on this exploration. Stay curious, and we look forward to seeing you in the next class.